So bean practices include no or reduced chemical inputs or disturbance from tillage, year-round living roots from either full-time ground cover or cover crops. Also, we suggest inoculation with beneficial microbes like what we produced in the Johnson Sioux composting bioreactor, or implementation to livestock with an adaptive multi-paddock grazing method. On this living planet, microorganisms have shaped almost every facet of development for the last 4.2 billion years. They've accomplished this by increasing system biodiversity and improving the efficiency of the capture, storage, and utilization of energy. As you look and see what's happened over the last 4.5 billion years ago, water hit this planet about 4.2 billion years ago. Bacteria very soon after. First novel adaptation was cyanobacteria with anoxygenic photosynthesis. These organisms allowed the capture of energy from the sun, the ability to store that energy and use it later, which has set up the whole system of life on this planet. Nitrogen fixation about 3.2 billion years ago. Oxygenic photosynthesis about 2.5 billion years ago, and this led to an oxygen-rich atmosphere. With oxygen in the atmosphere, fungi were able to evolve on the scene. The ozone layer built up about 1.6 billion years ago, and mitochondria evolved about 1.45 billion years ago. These are small organelles in a cell that allowed a 19 times increase in the energy use efficiency of these living systems. First single-cell animals about 750 million years ago, land plants 700 million years ago, and Gloma micata about 400 to 500 million years ago. These are the fungi that associate with plants and plant roots. When we look at the influence of life over the last 600 million years, there's significant changes that have happened on this planet in this short period of time. On this chart, you'll see the black line is atmospheric CO2 concentrations. They've been as high as 7,000 parts per million, and today they're down to about 400 parts per million. The red line is the oxygen, The red line is the oxygen content in the atmosphere. And you've seen it's gone up to as high as 30%, but today we're looking at about 20%. The blue line is the temperature of the planet. And you'll also see down here, there's extinction events, either meteorite hits or some of them we can't explain what happened, but there's a significant number of collapses in our environment. Started out in the Cambrian with the Cameron explosion. With the coral and shelled animals, see a significant decrease in atmospheric CO2. This is due to the calcium carbonate that is absorbed in the shells of these animals. Next on the scene here, you'll see gymnosperms, the progymnosperms, roots, leaves, and seeds developed. This produced another significant drop, all the way down to, in about 300 million years ago, a similar atmospheric CO2 concentration. This is when our cold seam development happened, and also peak oxygen. Conifers and ginkgos came along about in this era. In each adaptation that's happened on this planet, we get a higher ability to capture energy from the sun and store it. Angiosperms came along about 150 million years ago. Coral reefs had an influence in the development or the reduction of atmospheric CO2. And there was a cooling period about 53 million years ago. Mammals, ungulates, and grasslands all evolved in this area. So the grasslands, which developed these deep carbon soils in our prairies that we have farmed for about the last 200 years, was formed by these ungulates and the grasslands and their influence on this planet. So we're seeing that microbes are the backbone of every organism and ecosystem on this living planet. You as a human, your cell count, you have about 10 trillion human cells. You have about 90 trillion microbial cells in your body and about 1 quadrillion resident viruses. In a gene count comparison, you as a human have about 23,000 genes. Your gut microbiome alone has 22 million genes. So they outnumber you by 99.99% as far as their gene count compared to yours. What do these genes do for you? What do these microbes do for you as well? They digest your food, they do cell renewal, they comprise what we thought before was 60% of your immune system. Now it's more like 80%. They control your appetites and cravings. They do nutrient generation, vitamin synthesis. They detoxify carcinogens. They prevent allergies and skin diseases. diseases and asthma, and they turn on and off genes that regulate brain development, anxiety, depression, autism, arthritis, and emotional behavior. What happens when these microbial communities are disrupted through either diet-related issues, administration of antibiotics, or exposure to chemicals? We see significant increases in autoimmune diseases from these causes. Okay, let's look at a diet-related incident. When we have reduced the fiber in mice, generation after generation after generation, you see a decrease in the diversity of the microbiota in their stomach. Figure, okay, we'll just reintroduce the fiber back into the diet. That doesn't do it. They still get a significant decrease, generation after generation after generation. When you come in with a fecal matter transplant from a person or a mouse with a healthy microbiome, you have that microbiome back immediately, functioning properly. We're seeing over the last 70 years, significant increases in autoimmune diseases for multiple sclerosis, asthma, diabetes, and Crohn's disease. Crohn's disease, people can suffer decades with this. They can have resections of their intestines. Many people can even die from this disease. And yet they find when you come in with a fecal matter transplant from a healthy donor, some of these people can be cured within 24 hours. And they're seeing cure rates above 61% for most all of these diseases that you see here. So if microbiota transfers work in the human microbiome, can a simple inoculation of beneficial microbes help restore the microbiome structure and function in agricultural soils? First, we need to look and see how agricultural soils are designed by nature. You see, in a bacterial dominant system, a bare soil parent material, there's very little productivity. There's only about 90 grams of biomass per square meter. As we move from a bacterial dominant system to a fungal dominant system, like your old growth forest, you move up to increased productivity, up to 2,200 grams of dry biomass per square meter. Right now in our agricultural systems, we're here. We can grow weeds really well. We need to be in this range, fungal to bacterial ratio of 0.75 up to about 5 to 1. That will grow the row crops we want and the grasses as well. So 4 things have happened in our management of these lands. We have significantly altered the soil microbiome with our agricultural management practices. The soil microbial biomass has changed from fungal dominant to bacterial dominant. The soil microbiome diversity has also decreased. All of these have created a soil that has lost its biological functionality, decreasing the ability for the soil microbiome to supply necessary elemental nutrients to plants from the soil parent material. Another thing that we're seeing is a loss of soil depth. Since 1850, you can see we can lost soil year after year after year to now, in some instances, we're approaching a critical threshold. This is a threshold that you cannot grow a plant on. So we need to be very careful. We need to start rebuilding these soils. And I hope to show you a way that we can do this. This is nature's system of building the soil. Again, I was talking about the Great Plains, and it was the influence of the bison and the prairies, the grasses. The way the bison would come into this system, they were kept in tight herds by predators. They were moved along regularly. They were never really allowed to take the forage down all the way to the soil. They always left about 60% due to that influence of the coyotes and the wolves. The bison would also deposit dung and urine and saliva in this grazing process. This dung would be rolled into balls by dung beetles and placed into the ground, basically composting in place. It was like an inoculation event that swept across the prairies as these bison moved. So not everybody can have bison or cattle on their property. And we came up with a composting process. It was in a project with USDA, do something good with a dairy manure. And the composting process created a very good compost, a vermicompost product that seemed to, as I'll show you here, influence the soil, the ability of a soil to grow a plant and to have biodiversity. First of all, about the static, it's a static composting process. You need to keep it at 70% moisture content. No need to turn it. It's aerobic because we have 6 air columns up through the center of the pile. Do not let it freeze or dry out. Worms are added after the temperature goes below 80 degrees Fahrenheit. It takes about 1 year to reach maturity. And you need to follow instructions closely on the first try. No shortcuts on that one, but this is the link here to see how to do it. But it's very important on your first try to stick to the process. You can innovate after that, but you need to see how the process goes. When we look at the change in the bacterial community through the composting process from 159 degrees down to 60 degrees, you can see some of the bacterial phyla start very strong, but then they end up very small portion of the final compost product. Others start reasonably strong, they fade away, and then they come back to be a major component. And some of them start small and end up to be a major component in the end. This is a comparison of two composts made 10 years apart with the same process with different materials. The first one was dairy manure and leaves. The second one is just waste alfalfa. As you go through the composting process, you'll see there's a change in diversity in the pile. This is at 4 weeks. You see about 23 species in the top 80%. This is at 22 weeks. You'll see about 57 species. This is at 60 weeks. You see 99 species. So as you allow this compost to mature, you increase the diversity of the microbial community in that system, which makes it very beneficial when you apply it to your soils. So what do these microbes do for you? Well, nitrogen fixers. In this compost, you end up with a lot of symbiotic and free-living nitrogen fixing bacteria. You end up with phosphorus solubilizers. You have probably 40 years of phosphorus in your soil. It's just not available because of the soil chemistry. But when you bring these microbes in, they can free it up and make it available to the plant. You also have microbes that can oxidize the elemental nutrients out of the parent material. And that they can feed all the iron and copper and zinc and other micronutrients and let the plant have access to them. We also see phytohormone production. Some of the microbes secrete hormones that increase plant growth. And another thing, we see carbon and nitrogen cycle, which is very important in your system. You need to build a system that cycles both carbon and nitrogen so it's available to the plant. But what's most interesting in our research is we see quorum sensing. And quorum sensing is when a group of microbes get together as a community and express genes as a community that they cannot express when they're in a single organism. It's like humans, when they get into a community or work together, they can accomplish a lot more than they can individually. It's the same thing in the soil. We see a significant increase in quorum sensing as you improve these soils. We followed with a greenhouse experiment with Chile, assessing the influence of fungal-bacterial ratio on carbon and nitrogen partitioning. You see in the charts to follow, the brown will be the root carbon, the green will be the shoot carbon, the red will be the fruit carbon, the gray will be new carbon that's put into the soil, the blue is respiration carbon, and the orange is carbon that has been devoted to nitrogen fixation. When we look at the influence, as we change from a bacterial dominant system to a fungal dominant system here on the x-axis, you see an increase in productivity in the system. You see a lot of changes in the soil carbon and also respiration, but you see a consistent increase in the carbon that flows into the plant, roots, shoot, and fruit. Also, we track the nitrogen. You see again, as you increase fungal bacterial ratio, more and more carbon goes in, or nitrogen goes into your plant, root, shoot, and fruit. But you see that the nitrogen that goes into the soil reaches a peak about 3% organic matter. At that point, you see a decrease in the nitrogen production in the system because the system has the nitrogen it needs and it can cycle it. This is where we want to get you in your soils. You need to be above 3% soil organic matter to really get completely away from fertilizers. When you look at it in a pie chart, you can see in a bacterial dominant system, only 3% of the carbon that flowed into that system went into the plant. The other 97% went into the soil to have new microbes or respiration or fixed nitrogen. So it must be very important for a plant to devote 97% of its energy to put it into the soil instead of into its survival. As you move up this chain from fungal bacterial ratio of 0.04 to 0.84, 1.6, you see an increase of flow of carbon into your plant system. 17%, 21%, all the way up to 56% as you hit a fungal to bacterial ratio of 3.68. So it's a significant increase in productivity as you rebuild your soils. From the greenhouse, we went to a small plot test, looking at growing cover crops on a desert sandy plot. You can see in the background, this is the desert. It's never had agriculture on it before. And on this experiment, what we did is we grew cover crops. First of all, we inoculated the soil with 400 pounds of compost per acre rate. I'll show you, it's a dusting. It's not as a nutrient amendment, it's as a microbial inoculant. This is our second winter cover crop. We produced about 1,608 grams of dry biomass per square meter. Our first winter cover crop was about 800 grams. This is our third cover crop. And again, no fertilizers, just biology. And what we've done on this is we've grown cover crops, we've cut them and hauled them off, and then replanted and grown cover crop again. So we have not allowed any of the plant material to stay. We wanted to see just what potential the biology had for growing a plant. You can see this was 2,190 grams of dry biomass per square meter, a significant amount of productivity. There was 588 pounds of nitrogen in that crop. And this is, again, no fertilizers, just biology. Looking at the first 4 winter covers, you can see we started out at about 492 on the controls with no compost added. The green is the compost added. You see every cover crop following, we have a continued increase in productivity as you start to develop the biology in this system. From the first to the third cover crop, we saw a 4.5 times increase in photosynthetic capacity of this system. So the same amount of water, the same amount of sunlight, but you're getting more and more productivity. On this, we ended up with an annual increase of 3,186 grams. And if you remember back to the chart I showed you, the most productive ecosystem in the world was about 2,200 grams. So with the right biology, we produced half again more than the most productive ecosystem on the planet. And here we saw about 777 pounds of nitrogen per acre per year accumulated in this system. This is the spread rate. This is 1 square foot. It's just a dusting. And this was our initial amount that we started with. We next went to a field trial, a small field trial. And on the left-hand side, it was our control. On the right-hand side, it was this after 1 year of beam application. Again, no nitrogen or fertilizers added in this system. In the control, we saw 50 grams of dry biomass per square meter. In the beam, we saw 250 grams of dry biomass per square meter. We saw a 5 times increase after the first year in productivity, putting the biology back into the system. This is 7 years later. We saw 1,141 grams of dry biomass per square meter. So a 22 times increase in the photosynthetic capacity on this plot of land over a 7-year trial. These are some of the cover crops we grew in the summer on these same plots. On the left-hand side, you'll see a black oilseed sunflower. It's a mix with a couple other, I think there's a forage soybean in there. But black oilseed sunflower normally only grows about chest high. You can see in the back, some of these are 7 foot high, and the heads on them are much larger than the black oilseed sunflower. So we really don't know the capability of these plants when they're in the right soil. On the right-hand side is a Sespania crop. It's a legume, it fixes nitrogen, and it also associates with mycorrhizal fungi. The first season we grew this, it was 4 feet tall. The second season, it was 6 feet tall. The third season, it was 12 feet tall. And again, as the dynamics change in the soil, these plants keep getting better and better productivity. We followed up after the 7 years with a cotton crop, and we did about 2,200 pounds of lint per acre on this. That's about a 5 bale an acre crop. It's about $2,000 an acre gross profit on this crop with the seed and the lint. On the right-hand side, in 2018, we did a chili crop. We were getting 17 to 20 tons of chili per acre on this plot. When you compare those productivities to the norm in our area, we were double the productivity of the average production on cotton and also chili. Now, we look at organic, and organic cotton nationally gets about 750 pounds of lint. You can see if you can start to move towards a regenerative approach, a beam approach, you should be able to increase your productivity in your systems as you restore the function of the biology in these soils. We also saw increases in soil carbon. The rate of increase in the first 4.5 years was the 0.25% carbon increase per year in the top 30 centimeters or top foot. That's 10.7 metric tons of carbon per hectare per year, or 37 metric tons of CO2 per hectare per year captured. We also saw increasing ability of these soils to infiltrate water and to hold water. That first 1% increase in soil carbon led to a 5 times increase in the water storage capacity of these soils. So if you're concerned about losing water resources, if you implement cover crops, it's just the opposite. You are starting to build up your reservoir. You're building the ability for it to infiltrate and to hold it. This 1% increase ended up to be equal to about a 2.5-inch rain event. We also saw increases in micronutrient availability. This is a 20-month study in 5 sampling periods. We see 1,100% increase in soluble manganese and iron. Now in our soils are very basic. So these 2 particular elements are supposed to be decreasing in availability according to the physical chemistry of the system. But once you bring the biology back, we saw 1,100% increase in both of these, as well as magnesium, calcium, zinc, and copper. We also saw buildup of nitrogen, phosphorus, and potassium in these soils. We were able to get a field research opportunity with corn, looking at how this would happen, how this would benefit corn production. We implemented cover crops between commodity crops, and then we roll those covers and we planted the corn into it, and we would inject the biology, the compost extract, into the furrow at planting. We reduced evaporation in this system. We protected the surface. We also saw these cover crops offer a forage for soil microbes and beneficial insect communities, and also nutrients for your commodity crops. Over this 2-year period, we saw a complete change in the bacterial and fungal ratio from 0.009 to 1.35. A decrease in your bacteria, a 24 times decrease, and a 6 times increase in fungal biomass. We saw in this, the farmer 4 years previous to us taking on this project had been no-till. He was increasing his soil carbon at 1.26 tons of carbon per hectare per year. Big did it at 9 times that rate to 8.48 tons carbon per hectare per year. The benefits we observed 85 to 100% reduction in nitrogen fertilizer applications, average increases in commodity crop profitability of $120 an acre, increases in soil nitrogen reserves from 1,350 to 1,700 pounds of nitrogen per acre. So over that 2 years, the nitrogen we put into that soil with the microbes was worth $1,200 to $1,500 per acre. There was a 42% decrease in soil sodium concentration in the whole soil profile down to 18 inches, and reduced pesticide and herbicide applications. This profit per acre for our treatments was $86 to $122 an acre for corn, and $132 to $143 an acre for pinto bean. We got a cotton project in Turkey with Stella McCartney. We employed the same practices: growing covers, rolling the covers, planting into the covers. This is what they saw. They saw yields were 4.2 tons of cotton per hectare. The crops did well despite the government restricted irrigation and low rainfall. They only had 2 irrigation events. They completely got away from herbicides. They didn't have to do anything for weed suppression. They had 85% less fertilizer used with no yield impact. In fact, their yields were better with cotton fields that received 100% fertilizer. Big cost savings. The slate test on the right shows you much better aggregate stability in bean soils than from conventional fields. And they observed biodiversity in their soil that they had not seen since their childhood. They saw worms start to return to their system. They also implemented some of the compost on a plot of land that they said they were never able to grow anything. they're able to feed their families and all their friends' families vegetables this year. The Haggertys are doing a similar process in Australia. They are barley farmers, and they come in with a compost extract very similar to ours, and they do a seed coating, they do an injection at planting, and they also do a foliar application after the barley gets about 6 to 8 inches high. You can see the roots, the biology that's formed on the roots. They get about 3 tons of barley in a region that gets 1.5 to 1.6 conventionally. On the left-hand side, excuse me, this is after they've harvested the barley. You can see the stubble. I got a late fall rain, and in the area where the biology had been injected, a C4 grass they had not seen in 4 decades came up. So when the right biology is there, even the seed banks can be regenerated in these regions. This is Russell Hedrick, North Carolina. He's a corn farmer doing commodity cropping. He's introduced cows and sheep and pigs into the operation, plus cover crops. He's seen an 80% reduction in fertilizer application and no fungicides or pesticides in the last 6 years. His 6-year average is 185 bushels of corn compared to the county average of 110. They had a drought in 2015. They only got 4 inches of rain. He did 128 bushels of corn per acre compared to the county average of 9. So this is where it helps build your soil water holding capacity. So what we're doing is we're creating a positive feedback loop. The sunlight that the plants get, they're funneling that energy, that carbon energy, down into the soil to feed the soil microbiome. These microbes are extracting elemental nutrients from the soil parent material, fixing nitrogen from the atmosphere, and feeding it back into the system. A positive feedback loop, and each iteration that it goes, it improves the system. So this may sound like an infomercial, but this is the believe it or not portion. On the left-hand side, Paul Tranfield in Victoria, Australia, on his chestnut tree, he was only able to spray his grazing or pasture land on one side of the tree because the limb was too low on the other. You can see here on the left, this portion of the tree leafed out a full month ahead of the rest. And the chestnut on the left is the one in that greener portion, and on the right is the one where he didn't spray it. Patrick O'Neill in Alamosa, Colorado, they saw better production on their potatoes. This is Alicia Haft in Rockfront Ranch in California. This is her orchard with Johnson Sioux compost on their quince, with and without. And this is a vineyard, a Rick Kay in Puma Springs Vineyards, Healdsburg, California. You can see the Johnson Sioux compost on the left compared to an organic compost he gets in the area. Significant difference in the ability to grow a plant. So that's all I have for now. My suggestion is to start considering looking at a beam approach on your soils and align yourself with nature. This is a lot about observation. It's different processes in every region or every soil. So it's something that you have to develop a knack for observation to see how different things affect your productivity. But restoring the biology in these systems is key to bringing them back. But I like the question part better than the presentation part. More comfortable.
This is where the fun part happens, right? Well, we do have a lot of farmers in the audience, and so that's really exciting for them to get to connect with your information. Let's see, we're going to start at the top here. So, in Kentucky, I'm looking outside at like 2 inches of snow on the ground. What are some strategies, very practically speaking, that we could use to prevent freezing in the bioreactor? What do you do in these colder climates? What do you recommend?
It's good if you can find some way to insulate it or put it in a shop or a barn. We have people in Canada that put it in their barn with their cows. We've had people in Quebec make an anaerobic composting pile, harvest the energy out of it, feed it into a greenhouse area, and keep that greenhouse area warm enough for, well, they grow fish up there as well, but to keep the reactor from freezing. If you have a root cellar That's the ideal place. But just we found that freezing really sets this back. Since it is a vermicompost, the worms are the first to go, and you really need them. We're seeing that there are significant impact in the end product of this compost. I'll say vermicompost.
Yeah. And remind us, we did drop the link to your page with the, all the research and the YouTube channel and the video of how to make this bioreactor. So there were a couple questions for that. So everyone you'll have to scroll back in the chat there to access that. And we'll be sure to include it in the in-session details as well. Can you just remind us of the timeline, sort of, once you get it built, how long it's going before you can start using it?
We've seen that it really takes a year, and that's what we demonstrated in that change in diversity. And we see similar compost across the planet. This has been made in Australia, and I've done tests with metatranscriptome and metagenome analysis, and they're almost identical to a couple of products in Australia, a Johnson 2 bioreactor in Australia as well. So this can be made by anybody, anywhere. That's why we developed it really this way. We wanted to be people to be able to do this in a third world country or do it commercially in a parallel processing way in here in the US or in an industrialized country.
Great. Okay. So let's see. What are you thinking? I guess talk to us a little bit about your perspective on how cover crops are essential to building this microbiome, particularly for those farmers in the room that don't have livestock on the land, right? And aren't integrating livestock in their cropland. Can you just expand on that for a bit?
It's a system. It's what we're rebuilding, and not one component works by itself. If you put the microbes out there and don't have the cover crops, if you don't have that living root to funnel that energy into that system for them to build their community, it's like putting a cow out in a pasture without any grass. It's going to die. So you have to have a living root in the system. The cover crops, you can do this with cover crops, but as you see in our research, you can do it much faster once you bring the microbes into the system, especially that nitrogen fixation. That's really a key to starting to rebuild your soil and not have to import any nitrogen at all. With the proper biology, with the proper cover crops, and I go pretty heavy on legumes. I'm about 80% legumes on those cover crop mixes. And I'll bring in a triticale or a rye, something that can roll down well. But again, getting that nitrogen in the system, that's the major nutrient that we're really trying to get built up in your soil so it can start cycling. It's not just the fungal bacterial ratio. You have a whole suite of microbes in the soil food web. You have nematodes, you have amoeba, you have protozoa. All of these organisms work together to make this system function, to cycle that carbon, to cycle the nitrogen, and make it all available to the plant. So it's a system approach, and you have to look at everything as a system. And you don't absolutely need the grazers. This seems to take the place of them, and it allows, the aggregators are doing 20,000 hectares with this process. And the people in Turkey are so happy with this change in how they farm that they're also starting to go into Romania. People are asking them how they're doing this, and they're having to do all kinds of presentations and radio presentations as well from all the interest in Turkey. So it's an amazing process when you allow the biology to work.
Yeah, where it's sort of what you're talking about is really resonating with what John Kemp shared with us yesterday.
Oh, really?
Yeah, and so it's just so complementary how when you activate the soil biology, so much more energy is captured, right, and goes to feeding the soil even more and increasing the immunity, right, of that plant itself.
It's really a simple process. It just takes observation. It's aligning yourself with nature. I like that statement more than anything, because it gets you in that perspective that you need to be looking at this, how nature has accomplished this, and how you can mimic nature in your farming system. And it's very possible.
Yeah. So, there's some folks who are saying, where can I get some if I don't have one on my farm? That kind of thing. And we have Steve Diver on the call with us too. He mentioned that, there are a couple farmers potentially making some or selling small volumes that people could use, to start their own. So can you talk to us a little bit about like the broader network of folks, right, that are doing it? And yeah,
it's building up, but I still suggest that if you have trouble, start now. Get a bioreactor going. It'll be ready for planting next spring. There's nothing like doing it yourself. You start to develop an appreciation for nature and the earthworms. And as you see the compost, what we're putting down we put it down at 2 pounds per acre rate as an inoculant sprayed into the furrow at planting. That 2 pounds per acre, when you look at the number of fungal spores and bacteria that you're putting about 83 million bacteria per square foot and about 10 million fungal spores per square foot in that 2 pounds of compost. So it's a significant amount of biology that you're putting into the system. And the fungal spores, that's the nice thing about this composting process is you end up with a lot of fungal spores in that system because you completely use up all the energy, all the food resources in that compost pile. And of course, when a fungi senses that there's no more food to be had, they'll sporulate and the bacteria will insist. So we're putting out a significant amount of biology just at a 2 pound per acre rate.
So we have a question from Dr. Sean Lucas here at Kentucky State University. He is interested in if anyone's done the economic analysis of the system, not so much in terms of like the crop side of it, but just over time, is there an economic benefit associated with the methodology and an ongoing basis for that cropland?
Well, besides for a farmer being more profitable, building up his soil fertility, putting in from $1,200 to $1,500 of nitrogen per acre over a 2-year period, building up the soil carbon, having it as a vehicle for reducing atmospheric CO2, not having to use nitrogen. How far you want me to go?
Yeah, it's great. I think the question is probably getting at, is there published research on the economic impact of this?
I have a paper ready, waiting for the
We love that.
for the funder to approve it and submit it for
publication. So good.
But until that point, I think farmers get it.
Yeah, totally.
I don't know that they really need a whole lot of other data right now besides for this. To see they're more profitable, to see they're going to restore the fertility in their soils, they're going to rebuild the soil profile, because with these cover crops, you're leaving a layer of detritus. You're starting to build your soil up in layers, like nature did. When you look at the carbon dating on these soils, the younger carbon is in that upper profile. The older carbon is in the deeper profile. So you see, nature builds soils in layers, and rebuilding that soil carbon, not having any nitrogen inputs into the system, so you don't have any nitrous oxide going to the atmosphere or nitrates going into the water table, destroying our streams, our aquifers, the estuaries, all the reefs. There's a lot of, it's hard to put a dollar amount on all this will help. And then having people in countries where they've completely desertified all their agricultural, and they're having to leave their countries. This can help them start to rebuild their soils.
Yeah. I mean, there's no denying the incredible impacts. I think that having the research out there will be amazing too, the economic impact, because so many people, at a larger systems level or institutional level, once the economics are published and make sense in that way, then there's more rapid adoption. Yeah,
that's been the whole idea on these 2 projects we got. And we've only been in them. We're in our 3rd year in both of them. And these are the results that we've seen, that change in, well, productivity in the system. The farmer went out there with us and he was comparing his conventional at 256 pounds of nitrogen to the beam approach with only 15%. And we also had a beam with no fertilizer in. And you could not see the difference in the corn. We saw very close productivity. It was like 217 bushels of corn for both the conventional and the beam plus 15%, and a drop down to 205 for the beam only. And then the follow-up pinto bean crop, where the farmer lost money on the conventional approach because of the fertilizer that he applied. And he was more productive and more profitable in the beam plus 15% and the beam only. So there's a lot of economics here that we're seeing it in Turkey with their cotton. They're ecstatic with the process. They're going to be expanding, and this is one of the largest cotton farmers in Turkey. And they will be expanding, as I say, they'll be expanding into Romania with barley up there.
All right, we have a couple questions about the details again here. How much land would the contents of one bioreactor cover for in terms of the recommended application?
It takes about 2,000 pounds of wet material to fill a bioreactor. You end up with about 700 pounds. At 2 pounds per acre rate, that's 350 acres.
Great.
So it goes a long way.
Yeah.
Again, quality, allowing it to mature, allowing nature to do what she does to that compost to get it to the point that it's very good for your soils.
Mm-hmm. And another very detailed question: do you filter out the worms before you're distributing the compost, or what's your recommendation there?
Well, yeah, in the making the extract, they do take a beating. Some of them survive it. There's also an extractor video at California State University, Chico, in their Center for Regenerative Ag. There's a poor boy extractor that I put together that you can salvage some of the worms in the process. Yes, a lot of them, they don't make it.
Yeah. And I guess there's another question from Jessica. Do you have an opinion or any results, thoughts on the incorporation of biochar into the system?
Well, what I try to give the perspective of is we're rebuilding a living system. And the carbon that we're putting in by growing it in is a functional carbon. It's a carbon that's in the shape of a microbe or a fungi. And the biochar is not a living carbon. So it's great. It's a great hotel because it's very cavernous and it allows the microbes to populate. But as far, there's a limit to the amount of biochar that you put on a soil and not affect the productivity. We've seen that in several people's other people's research. But as I say, it's a different biology. It's a different perspective. It's a living system again that we're trying to tell people about.
Yeah. So here in the Kentucky region, we have a challenge with Johnson grass. And a lot of places have challenges. So there's always this question of how can you outcompete it, right? How do you manage it? Could you outcompete it? And so I guess the question is just broadly, have you seen in beam systems used with cover crops, have you seen the system able to outcompete, some of these more challenging weed pressure?
Yes, we have seen with the purple amaranth and its herbicide resistance, people that are employing these cover crops and rolling them down and they smother, and they're actually able to get away from having to use any herbicides. So Johnson grass may be a little tougher, but if you can come in and have that cover, you will start to be able to work away from it. I mean, the people in Turkey, they had 23 passes in the field to take care of weeds and insects. They didn't have any this year. They completely stopped their weed problem and the beneficial insects kept the ligas away from the cotton. So, there's a lot of benefits here, besides for weed suppression. But, you would just have to try it and innovate and see how you can take care of it. Yeah, I know Johnson grass. We had it on our farm in New Mexico, and it was an issue. But I was an MPK junkie till I got along this vein. So
yeah.
I'm about 15 years.
I was going to ask, and when did that start? You're 15 years into it?
Yeah, I'm about 15 years sober.
What was it? So let me ask, I mean, we have some time. Let me ask, what was your aha moment?
Oh, well, okay.
I mean, no pressure. Was there an aha moment? Sometimes this makes for
I have to admit, this completely took us by surprise. I was a contractor for 30 years before I went back and got into academia. I built commercial and residential buildings. And it was the influence of the classes I took, the professors that I got in with, their research. The second day on the job at the university, I got handed a project, do something good with dairy manure. The people that had it a decade before me had concluded compost was bad for soils. I looked at their data and they were right, because the way they were making compost, it was a very saline compost. It was actually very hard on a soil or a plant when you applied it. Our process was totally serendipitous. I started with the windrow approach. My wife, Wei Chen, sitting here, she said, "I'm tired of this dirty laundry. We're going to do something different." So it is, behind every mediocre man is a great woman. That's what it is here. A lot of this is because of her influence and her observation. And we developed a composting process that was no turn. And lo and behold, but it reduced the salinity of the compost. Again, we see it in that field in Arizona for the corn, that 42% reduction in sodium in that soil pretty much indicates that the biology is doing something. Either it's locking it up so it's not soluble, but a 42% reduction in a saline soil that uses saline water to irrigate is significant. And it was the same thing in the compost pile. We saw that reduction in salinity from about 33 to 40 decisiemens down to less than 3. Wow. Anything above 10 damages a plant.
Wow. Oh, well, thanks for sharing that story with us. It's always fun to hear the behind the scenes coming.
Well, and it's been like that on everything. It's always that side road, that anomaly. What's going on here that has spurred our research? We didn't go the common path. I wasn't. I'm a molecular biologist. What would a molecular biologist be doing in agriculture? Until recently, we see the influence of the microbes on the human genome. So,
yeah, always be curious, right?
always be observant, always be curious.
Yeah. Well, we have just a little bit more time. A couple questions here in the chat. One question from Phyllis is bean compost allowed in certified organic operations? What are you hearing from organic farmers about how they integrate this on their farm in line with regulations?
I think in every state but California it is, because it's a 1-year process. It's a vermicom composting process. So they allow that time frame. If you keep it in the moisture content, it does degrade everything. Now in California, with worms in the compost, they have an issue. They call that livestock. So you have to wait a certain time after you apply it in California. But you're applying it at such a small rate.
I think it would be the same here. It's considered manure, right? Worms the FDA considers worms livestock. And so worm poop is manure. And so it's that same 90, 120 day interval. that farms would want to follow, both for food safety and for the regulations.
Which is a shame, because when you look at the microbes in this compost, there's almost every pathogen known to man. We're so sensitive on the test, we can tell who is there. So we do need to change our perspective on nature and soils and microbiomes. And I think it will happen. I mean, I think probably what promoted all these rules was people doing things wrong and not composting things completely and putting things out that they shouldn't have been putting out. And I think hopefully there'll be a change to be more open to vermicompost, because it is so beneficial. In Australia, they're seeing so many benefits. In Belgium, they applied this as a seed coating and they doubled the productivity on their cover crop. So there's too much to be gained from this, that maybe some rules need to be changed.
So funny you should mention that. Our last session yesterday, which was a panel on food safety, it was brought up that FDA has an open survey right now open for comment on some of the food safety rules. So there's an opportunity, right, to share. Let's reconsider, this vermicomposting thing. So we'll send you that link. And once we have that available, I don't know, I don't have it right at my fingertips right now. But it would be great to share your experience, right, and your research into that conversation. So, okay, let's see. We have time for one more question. I think let's go with a question from Mary Kate. She's curious if you have any reflections on Korean natural farming practices in relation to BEAM. Are there key differences you would recommend, or are there key differences, or would you recommend them equally?
I'm not really too up on the Korean farm, as far as I know. They use rice, and they do a fermentation. I think mine is aerobic. And that 1 year process, I'd love to look at the biology in their system and see how it would compare to this compost. To see identical compost in Australia and the ones I make here in the US would say that there is a common endpoint when you compost aerobically in these systems with the presence of worms. There's a common product that comes out, because two of the four in Australia that are identical to mine are a commercial product over there. One is a compost made by a lady that has a different process, and another one is an extract. So,
yeah. So I guess there's another question asking about the actual. Have people used it as a foliar spray at all, or are they just applying the actual thicker material on top of this? So, talk to us about the different types of application you've seen and how it compares.
Well, we've seen, of course, we inject into the furrow at planting, do seed contact, do a seed coating, do a foliar on pastures. That was the one in Australia that Paul Transfield. If you spray a foliar, I've seen it completely stop any tomato wilt. I've seen it applied to a tree that had weeping wounds, actually fluid coming out of those wounds. I've just take the compost and pasted it onto that wound. It stopped that. There's a lot of things that
lots of applications, all the applications.
Yeah, just use your imagination and try it. I mean, it's so similar to what we're doing to our stomach microbiota with those fecal transplants. We're restoring that community into that system, restoring that balance. I don't see any end to the benefits if you can do that in a system that's broken.
And David, in our early conversations with you for this conference keynote, we asked if you've worked with anyone in Kentucky, and we determined, I think at the time, that there weren't any active BEAM facilities, compost reactors here in Kentucky. But I guess I want to just leave it open. If anyone on this call is interested in setting one up, we'd love to know. I'm sure David would love to know.
Yeah, we'd love to see that. We're working, trying to get one going in Iowa to to have farmers start doing this and to take data to see how these things, how it works in your region, in your soil, in your climate. You know, we've been pretty reduced to a desert climate here in New Mexico and Arizona and California, the 3 that we have going. And Turkey is not much different, but we see people in British Columbia and they're having success with it. We see people in Belgium, they're having success with it. Africa is it's in Africa. This process is in 60 countries that we know of right now. So it's pretty well worldwide. We're just waiting on data.
Yeah. Well, if anyone's interested, let us know. We'd love to connect you with some grant opportunities to help make it happen. And then we'd love to help tell your story, right?
I'll put you to work over there, Brooke.
Yeah, and help share, what's happening on your farm and what you're seeing on your farm with other farmers in the region.
It's very important that we get the farmers involved and they start doing quorum sensing, working together as a community, comparing what they're seeing. That's what Gabe Brown does with his neighbors. They dare each other to do something crazy every year. And then they look and see what the product of that is and what direction they need to go. Yeah. So it's important we work together like the microbes do as communities, helping each other.
Yeah. Oh, well, we are at the end of time. So I want to say thank you so much for all the inspiration and the incredible research you do and that you're sharing with so many farmers out there.
And thanks to all the farmers on this. We need you.
We do.
Badly, we need you. I realize this. I'm in it for the food, okay?
All the good food, yes. Well, David, we're just so grateful to you for joining us today. And we look forward to staying in touch, okay?
Likewise.
Take good care.
You too. Bye-bye.
Bye.